Hello. Oh. Oh, yeah. Okay. Test. Oh, no. Oh, okay. That's annoying. Okay, I'll keep the intro short. Welcome everyone, and thank you all for joining us today. My name is Terrence. I'm one of the organizers for this year's Quant Sem for the Punt Lab. I'll begin with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. Now to introduce our speaker, Autumn Maust is a PhD candidate in the School of Environment and Forest Sciences, studying the intersection of forest entomology and fire ecology. Her research explores the effects of mixed severity wildfires across temporal scales on pollinating bees, beetles, and flies in eastern Washington. She uses both field-based and molecular models to answer questions about pollinator community composition, bee fitness, and plant pollinator networks. So Autumn will take questions as we go during the talk. So thanks everyone for coming today. Big thank you to Autumn for sharing your work with us today. And with that, it's my pleasure to turn it over to Autumn.
All right. Thank you, Terrence. Hopefully folks on Zoom can hear okay. My name is Autumn Maust. I'm a PhD candidate in the School of Environment and Forest Science over in CEFS. And I'm going to be talking today about my work thus far, researching how wildfire shapes pollinator communities across 3 mixed severity burn sites in eastern Washington. So first, I'm going to back up and just do a quick introduction about pollination. For a lot of folks in this room, I recognize this is probably review, but I just want to make sure we're all starting from the same spot. So there are sort of a suite of insect pollinators that help to pollinate on an ecological scale. Those include beetles, flies, butterflies, wasps, moths, and of course, bees. And these pollinators are critical for food security for us as humans and have a huge economic impact as we're seeing global declines in insects and pollinators on a global scale. They're also responsible for the reproduction of over 75% of angiosperms. So a lot of our plant diversity is reliant on our pollinator communities being healthy. And they are a food source for other trophic levels, such as birds, and engage in herbivory. Think of a caterpillar for butterflies, eating milkweed. So that's an important role they play as well. But usually when we're talking about pollinators, as we all know, we're usually talking about bees, and there's a couple reasons why that is. The first is that they're the most abundant numerically on the landscape. There's just more of them compared to other insect pollinators. They also exhibit flower constancy, so they're more likely to go from flower to flower within the same species, as opposed to switching between species, which makes pollination more successful. They have adaptations that help pollen stick to them, like pollen-carrying hairs and pollen baskets, and their offspring is the only insect offspring that will consume pollen and nectar throughout its entire life cycle. You can juxtapose that to say something like a caterpillar eating milkweed, as opposed to eating nectar or pollen in its earlier instars. So usually when folks think of bees, they think of this, right? Like Apis mellifera, the European honeybee, out on farms and folks beekeeping. We all love the honeybee. It's very cute, but that's not what we're talking about today. The European honeybee, as the name implies, is from Europe and is not a native pollinator. Instead, my work is focused on native bees, which include resin bees that carry pollen inside their bodies, sweat bees, leafcutter bees, mason bees. There's some really cool cuckoos of our native bees, which don't pollinate as much, but do steal nests and still consume pollen, which is an important interaction to keep in mind. We have longhorn bees, mining bees, of course, bumblebees are probably the most familiar to folks in this room, and so many others. Over 895 is the current estimate native bee species in Washington. But I will say that a lot is still unknown about the native bees that we have here and the native bees we have in this country in general. And that's maximized on a global scale where there's not as much research occurring. So when we think about our native bees, we can kind of separate them into two nesting guilds, ground nesting and cavity nesting. And so just a brief look at the life cycle of a ground nesting bee. Here's the life cycle for Andrena, which is a mining bee. And it starts off as an egg with a pollen pod that's laid sort of underground in these tunnels. And that egg will develop into a larva, overwinter as a pupa, and emerge as an adult where it then forages. We see it on the landscape. And then each individual queen is going and making one of these nests on her own. They're not social like honeybees are. There are some social native bees like bumblebees, which nest in colonies, but for the most part, most of our native bees are solitary. And we also have cavity nesting bees, which find hollow reeds, holes, stems on the landscape. Old beetle galleries are an important nesting resource for them. And they nest in a similar way. They lay a cocoon at the back, and then they move forward, laying females in the back and males in the front, so the males can emerge first and be ready to mate. And same thing, the egg will develop into a larvae, which consumes the pollen provision and develops into a pupae to overwinter, and then emerges as an adult bee in the spring. With that, let's move on to fire ecology. So my research is taking place on the Okanagan National Forest land in eastern Washington. And in this area, the fire regime is historically a short-term, low-severity one, meaning pre-colonial settlement, basically. These small, frequent fires were coming through the landscape and sort of cleaning up the understory. And these fires were a result of both indigenous burning and natural ignitions through lightning. However, post-colonial settlement, we started fire suppression, which is like the Smokey the Bear era, no forest fires, and that sort of lasted the last 100 years, which has resulted in a lot of fuel buildup on the landscape. So this is a ponderosa pine forest in this photo, and you can see that there's a lot of sort of young trees very close together. They're mixed conifer, so it's not just ponderosa. So where we would like to see mature ponderosas in clumps with park-like open spaces in between, we're instead seeing a lot more landscapes like this, which result in higher severity fires when they come through because there's more fuel. And this disturbance has a mixed effect on bees. Folks, to my knowledge, haven't studied post-fire bee communities in Washington state, but from what we know from Oregon and Colorado, fire is generally good for bees. Of course, there's the immediate disturbance of bees sort of burning up or being displaced, but mostly fires are coming through cleaning up the landscape. They're increasing the availability of floral resources. They're increasing those cavities for cavity nesting bees by hollowing out stems and resulting in more dead and downed material. There's more light hitting the soil, so it's a bit warmer. And so for all these reasons, typically post-fire bee communities are improving, which leads me to hypothesize that if we look at several burn sites across this landscape, we can sort of find this sweet spot post-fire where those benefits are maximized and then maybe they decrease over time with succession. So I mentioned our ground nesting bees, and some folks have studied the effects of fire on ground nesting bees. This is one cool study that I just love to highlight because I think it's really neat and specific. And it basically shows that ground nesting bees that nest below 10 centimeters in the ground are able to survive heating by wildfire. So these ground nesting bees are doing okay when those fires come through. Granted, this is in like a low severity fire scenario, but still good news for our ground nesting bees. From the same study, this graphic is just showing which stage of their development they're surviving the most. So I mentioned there's the egg, the larval, the pupal, and the adult stage. And we can see the highest levels of survival from soil heating by wildfire in the pupal stage, meaning that late season fires, when bees have developed into this stage, are more resilient to fires that come across the landscape. This could indicate to us that earlier season burns or increasing durations of the fire season could be really detrimental to bees because they're in a more vulnerable life stage, specifically the egg stage. However, when we think about our cavity nesting bees, because they're on the landscape and it's a little harder to A, find them, and B, rear them in a lab, we don't really know a lot about how they're doing post-fire. So my research question is, how, if at all, does wildfire affect pollinators? And can we detect significant changes in community composition, fitness of those cavity nesters specifically, and floral hosts across burn ages? And to answer these questions, I have 3 objectives. The first is to quantify seasonal changes in bee species abundance, richness, and diversity at 4 field study areas representing different intervals post-fire, ranging from 1 year to over 30 years old. I also plan and have begun assessing the development and fitness of cavity nesting bee species across successional stages post-fire. And finally, using next-gen sequencing on pollen collected by native pollinators to potentially reveal high priority plants that we can emphasize during restoration to support pollinators in post-fire landscapes. I mentioned I'm doing my work in the Okanagan-Wenatchee National Forest. It's sort of indicated by the star on the map of Washington in the right. And here are my burn scars. So I have the Chelan Complex fire, which burned in 2015, and this orange perimeter here. The Cougar Creek fire, which burned in 2018 as the red in the bottom corner. And then in 2022, we added a field site, the 25 Mile Creek fire, which is the yellow perimeter, and actually burned in 2021. We also have a couple field sites that are outside all of these fire perimeters intentionally. They're right here underneath the Chelan Complex fire. And this site hasn't burned since 1968. So could provide like sort of a comparative site where fire suppression is still dominant on the landscape, and the results of fire suppression are still dominant. This land, it's important to note, is the native land of the Chelan, Okanagan, Yakima, Confederated Tribes of the Colville, and Cayuse, Umatilla, and Walla Walla nations, who, like I said, historically managed this landscape in a way that allowed those low severity fires to come through in the way that they naturally would. And so I just want to acknowledge that. If we take a closer look at my field sites, there are 2 plots for each objective, between objective 1 and objective 2 on the landscape. Don't worry too much about the photos right now. I'm going to talk more about them later. But just know that there's 2 plots for objective 1 where we're looking at community metrics. And over 500 meters away, there's 2 plots in each burn scar for objective 2. The 500 meters is important because the average bee flight range is 500 meters. So this ensures that we're not resampling the same community. Even though my sites look really close together on this map, they're actually far enough away that they're separate communities within the same burn scar. We've also monitored other explanatory variables that could be influencing what bee species we're seeing on the landscape, including vegetative analysis of the basal area, so the amount of area within 50 meters of each plot centroid that is trees, as well as floral abundance. In floral abundance, we've measured along transects and randomly placed 10 by 10 meter plots every 2 weeks. Over 40 species of plants have been identified on the landscape. We've also looked at topographic variables, the fire history, so how severe was the fire, what was the soil burn like, how big was the fire, etc. And then, of course, we're separating data by site, plot, date, and trop type, so we can look both through time and across burn scars. And here's some examples of some of the plants we've seen on the landscape in these places. To take a closer look at that, here's one of my sites through time. This is the Cougar Creek fire back in 2021, so my first field season. And you can see that in April, it starts off being dominated by airleaf balsam root. It's quite abundant. Come May, we have a bit more diversity. So we have some lupins, they're a bit hard to see on the screen here, coming in. By June, the snowbrush ceanothus is blooming. And then by July and August, we have what appears to be a pretty steep drop off in both floral resources and water availability on the landscape, as well as air quality. By August, it's usually pretty smoky because other fires are usually burning in the area. So this is what it looks like in 2021, but we do have localized temperature probes at every site because, of course, each year is different and increasingly unpredictable under climate change. So in 2021, my April sampling looked like the bar on my left. But in 2023, this past summer, it looked like this when I set my traps the first time. And when I came back 24 hours later, it looked like this. So a little early this year, a little late snow. important to keep track of temperature for those reasons. So when we take a look at burn severity, I mentioned that my sites exhibit mixed severity burns. So we'll talk a little bit about what that means. That mixed severity is a higher severity than what historically would be on the landscape, which was usually consistently low severity. And when we look at satellite-derived severity maps, we can see that it's sort of like a patchy burn with some pockets of high severity, some of low, some unburned, across all three burn scars. But this satellite-derived data isn't really the best for localized studies when we're really concerned about plants on the ground. These are generated based on reflectance, and you can imagine a blackened terrain that used to be grassland would look like quite a severe burn because there'd be nothing left, but a complete forest that had complete crown kill and burned right to the ground would also appear black or like it was a high severity burn. So it's important to take into consideration what the vegetation type was that was burning and not just rely on these satellite-derived severity maps for studies like this that are concerned about vegetation and the local scale. So instead I'm using composite burn index, which does just that. It takes into consideration what the landscape was pre-burn, and it's run through Google Earth Engine, and you sort of get these burn severity maps that are a little more fine scale and have a standardized method for comparison across sites. So it gives you an index 1 through 3. And here's two maps just of the Chilean complex site and the Cougar Creek site. I haven't analyzed my data yet, and I haven't actually run the 25-mile creek site yet because it was added later. So circling back to my objectives, when we're trying to create diversity metrics for these communities across post-fire burn sites, my hypothesis is that native bee abundance, species richness, and species diversity will vary within study areas and change over time due to decreases in habitat quality, and also that burn age and burn severity will be the main drivers of intercommunity variation. So sort of assuming that this time since burn actually does play an effect on what we're seeing in terms of community composition on the ground. And to test that, I'm using both blue vein and pan traps. So we are destructively sampling. I know I went on and on about how bees are decreasing on the landscape, and then you're saying, why is she destructively sampling bees? And the reason is there's not a solid collection or database of what bees are even here in Washington. So identifying them is extremely difficult, particularly on the wing. So when you're doing catch and release projects, it's easier to do with larger bees. But for smaller bees, it's a bit more difficult if you do want that species level data, which I think at this point in what we know about pollinators is pretty critical. So when using these traps, there's a couple of things to keep in mind. One is that there's collection bias, so different traps attract different species better. So we've used both. And also that what you catch can vary based on how high you set the traps and what colors you use. So we've kept that consistent across all 3 years of data collection and across state. We left the traps out for 24 to 36 hours every 2 weeks from April to August in 2021, 2022, and 2023. And as I mentioned, we added another site in 2022 after the 25-mile creek fire burned. So we were able to get in less than 1 year post-burn for that site. Then we take the bees back to the lab. They're dried, pinned, and then identified to species where possible using taxonomic keys, of which these are a few. I should have changed this so that it wasn't the Latin names, but basically there's quite a few keys we're using for bees, Hymenoptera, and then we also catch beetles, flies, and butterflies, and I'm trying to identify them at least by morphogroup, but to genus where possible. Here's what we found so far. So again, Hymenoptera is bees, and over time, we've caught quite a few, crossing over 117 species. And you can see an increase in abundance through time here. But I would say to use caution when reading into that. We'll have to account for imbalanced sampling during our analyses, but we did add a site in 2022, and we did sample 1 week in advance in 2023, hence the snow. And so just because those numbers look like they're increasing, doesn't mean that the number of bees in the landscape was increasing, even though that would be amazing since I'm destructively sampling, and it would be great to see that there's more bees, even though I'm taking some away. I still have a lot of analysis to do on these sort of raw numbers. when we look at the abundance of those bees through time, we get graphs that look like this. Some important things I like to point out are that I delayed the deployment of the blue vein traps until late May, both years, and actually all 3 years, because they are known to sample bumblebees quite effectively, and we don't want to destructively sample bumblebee queens where we can avoid it. So just pan traps prior to those dotted lines. And additionally, the spikes you're seeing at the end of the season, I don't believe are reflective of more bees being present on the landscape late season. Recall back to that graphic of my sites through time. By the end, there's few other floral resources on the landscape. And so I have a feeling that they're disproportionately attracted to my traps because there's so few floral resources on the landscape. And in terms of destructively sampling, that's actually not such bad news because by that point, they're probably done laying their nests for the following year and they're going to die soon anyways because it's the end of the season. So in terms of my analytical methods, I just have sort of an infographic of what I'm planning to do for analysis. This is still very much in progress as I'm not planning to publish this until probably late 2024. But I'm planning to use a generalized linear mixed model with both fixed and random effects. So looking at pollinator abundance, species richness, and diversity as a function of time since burn and fire severity, and where possible in the model, given limited power that I anticipate having, also including these vegetative, topographic, fire history, and collection variables as well, with the random effect of space and time so that we can determine differences across burn sites as those burns age. And then to address the second hypothesis, I'm planning to run a permanova with both, again, fixed and random effects of similar things to analyze intercommunity variation. I've done just quantitative analysis of the 2021 data using non-metric dimensional scaling just to sort of visualize what we have so far looks like. And you get a graphic like this for folks who haven't looked at NMDS before. It kind of just visualizes your data in a 3D point cloud that doesn't really have axes. And what I notice about this graphic is that our 6-year burn and our control that hasn't burned in a really long time seem to overlap in terms of polygon area pretty strongly, which could indicate that they are more similar, where the 3-year burn is a little shifted outside, could indicate that there's a difference there. Of course, we need to do a qualitative analysis before we can definitively say that. But adding in the 25-mile creek, which is even younger, could sort of highlight that pattern a little more clearly. And it's just a shepherd plot, letting us know we can trust this infographic despite its ambiguity in 3D interdimensional cloud space. Last thing for objective 1, when I run an indicator species analysis on the bees that we found in 2021, we do see that some bees are strongly associated with different sampling dates, which isn't surprising. We know that there are early season bees, late season bees, but this sort of corroborates that. And most of those strong relationships were seen sort of in June. This graph isn't, again, quantitative, but it does sort of let us look back at these lovely bee photos from Sam Droege and get a sense of what we're seeing through time in terms of highly associated temporal hymenopteran relationships. I can pause there and see if anyone has questions, just because I know I've been talking a lot before I dive into objective 2.
So do you have a quantitative way to measure floral diversity to look at that quantitatively, or do you figure that out?
So this is a good question. Quantifying floral diversity has been a huge challenge for this project, mainly because there's not really a standardized way to do it. And bees are foraging over 500-meter radius, basically in a circle, which I am not quantifying all of. So I have these randomly placed 10 by 10 meter plots that I'm surveying in transects every 2 weeks, but that doesn't mean that I'm surveying what the bees are even visiting. I'm just surveying where they were known to be foraging because that's where they were caught. So that's sort of how we're trying to quantify it and to keep our fingers on the pulse of what's blooming when on the landscape and how many floral resources might be available, because we do know from those analyses that late season, there's not much. But my third objective using next-gen sequencing on the pollen the bees have collected is a more targeted and hopefully more accurate way to determine exactly which plants they're seeing and how many, and then we can integrate that data that way with a bit more confidence. That makes sense. Cool. Any other questions so far? Great. Yeah.
Does the bee lay its eggs in the nest that it stated, or can it make find a new nest within that 100 radius?
Oh, good question. You mean the nest that it emerged from?
Yeah.
Does the bee relay its eggs in that same nest? All of the females that are emerging from their, I'll call it their like pupal origin, make new nests. So there's say 3 males and 5 females that come out of every nest. They'll mate or mate with other individuals. And then all 5 of those females will go create their own nest for solitary bees. A lot of times these solitary bees, though they are solitary, they do like to nest in an aggregate way. So they'll nest near other females of the same species, but they don't work together to raise young in the way that honeybees do. Yeah, Madeline.
Do you know, so I know like the blue vein and Pantraps is supposed to be like very much flower mix. Do you
Yeah.
know if there's like evidence for like in like there's drought or like water limitation, if you're gonna see more regardless of like total abundance, but it's like they're also drawn to that as like a water source, and so that you might be seeing more in like drought or heat conditions while visiting your or getting trapped in your pan and blue vein.
I think about this a lot, especially late season. And I know that there are some things that are probably attracted to the water in my traps, namely wasps, because they would not be attracted to the visual lure because they don't typically visit floral resources there after other insects. Could they be after the insects that are dead in my traps and looking for a snack? Yes. So it's hard to kind of parse why I'm getting bycatch that's non-pollinator related. But I don't know of any studies that have looked at like the effects of drought on trap catch. I am unsure.
Like I guess all 3 of your sites are experiencing the same temperature at the same time.
They are.
Kind of, and they're close enough, so it's like they're all getting a heat stroke, they're all gonna be.
They are, yeah. And we could look at that because we do have those localized temperature probes at each site. So specifically, I don't remember exactly when the heat dome was, 2021 or 2022, but I thought it could be really interesting to look at that specific period just to see how that affected the pollinator communities that we were able to sample, because I was doing field work during that time and it was certainly quite a bit hotter than normal. So something we could look into with the data. As you can tell, I haven't really finalized any of my analyses yet. So I think once that happens, we can sort of play with exactly what questions we can ask and different things we can do with the data set. Yeah.
I know you focus on pollinator, but it's just like a bit of comment that I would be interested to know like others like Leptoptera, Coleoptera, like what they're like what the population dynamic like post fire. So that would be awesome.
Yeah, definitely. That's why I want to like keep track of the bycatch, even though just identifying the hymenopterans has been an enormous undertaking and kind of full-time job. But I do think that it's important to acknowledge that they are also there and they're also contributing to pollination. I will say, and I haven't looked, but typically in entomological research, lepidopterans, butterflies are one of the most highly studied insects. So it's possible that someone's already done that work. And I would encourage you to look into it if you're interested, just because butterflies are like the big, charismatic, beautiful. Most insect collections have a majority Lepidopteran specimens, and most folks who are into entomology recreationally will target that that class, that order. So it might be out there, but I haven't looked very closely. More focused on bees. Okay, any other questions this time? Yeah.
One quick one. So bees, I'm assuming, are very visual predators or visual creatures. Given that they are looking for flowers and nectar. So in years when the smoke, even in areas that weren't directly experiencing burning, when the smoke is really high, do you think that that impacts bees' ability to find a trap, or is it improving visual direction?
Yeah. That's a really good question. And a lot of folks have looked at the effect of wildfire smoke on honeybees, because in an agricultural sense, that's really important. Obviously, we use smoke to disorient bees and kind of make them drunk when we want to go into the hive and collect honey. And so certainly quite a well-known effect on honeybees. They have a hard time finding their way back to the hive box when there's wildfire smoke. In terms of native bees, I would imagine that the pheromones are disrupted in the same way, or their ability to process the pheromones are. I don't know that anyone specifically studied that because it's very difficult to find native bee nests on the landscape. And it would be, in order to test that, you would need to bring a nest into the lab and then sort of like pump wildfire smoke in and then determine how well they are able to forage beyond that. Or maybe Madeline has a better idea. I know you all do. Okay. Maybe some like, I thought that there was maybe some like GPS wild bee tracking that was happening with Bombus, where you could maybe look at that on a landscape scale a little more effectively. But otherwise it would be really hard to do an observational study on the effect of wildfire outside of a lab setting. But it's a good question, a good thing to keep in mind. I think that my traps are so bright that even if they were super disoriented, they're having no trouble finding their way to my traps. Late season, at first when I started doing this study, I was kind of like, whoa, that's way more bees than I wanted to collect. But other bee biologists have assured me that late season, they're not as critical for pollination. There's nothing to pollinate and they've already reproduced. So if we are gonna overly sample, say, it's great, it's better to do it late season. So yeah, definitely. Okay. Thinking about my second objective, where we're focused on cavity nesting bees specifically, I'm asking if at the individual species level, cavity nesting bee oviposition and larval development would be highest, maybe 2 to 3 years post-fire is what I'm guessing. And that's based on the fact that I know that there's increased cavities post-fire, and sometimes it takes bark beetles a little time to come through and completely kill trees and make those galleries post-fire. And so after 2 to 3 years, when the bark beetles have kind of done their thing and everything has settled and cavities have been created where they're going to be created, are we seeing more cavity nesting species? And here we're using fecundity as a proxy for fitness. And so my computer might die. And so we're looking at the number of eggs that successfully develop into pupae as a proxy for fitness here. By looking at them. Should I take the silver one out? We could lose everything. Okay, it was a gamble. It worked. All right, great. So my methods for this chapter, I sampled over a period of 8 weeks, and we deployed these trap houses that you can see on the side. They're real fancy. They're made of PVC with paper tubes drilled on kind of like a small piece of wood and put on a tea stake on the landscape. So the idea was that if we created artificial cavities on the landscape, because it's often a limiting factor for cavity nesting bees, would they nest in them? And to hedge our bets, we released a native bee that's used agriculturally, Osmia lignaria, which is the blue orchard mason bee. So this is kind of used in the same way Apis mellifera, or the European honeybee, is used, but it is a native bee. So after the bees sort of went and foraged and they nested in the tubes, we counted, we brought the tubes back to the lab and then counted the number of tubes that were successfully nested in, number that they attempted to nest in, and number that were bare. And then we also quantified how many of the eggs were able to successfully develop into pupae for that metric for fitness that I mentioned before. So when we open the tubes up in the lab, this is kind of what we see on the photo on the bottom there. You can see the paper tube is opened and the bees, as we saw before, sort of lay their eggs in this partition and partition each egg with a pollen provision. And when the eggs successfully developed into pupae, we'll see that the pollen provision has been consumed. An example of the pollen provisions up there on the top. And we'll see a pupal case like we do on the top there. We'll also see a whole bunch of other things like mites, which you can probably see, and kleptoparasites occasionally. So these are beetles that lay their eggs in the nesting tubes and then in 2021 emerged before I broke open the nesting tubes and ate their way through all of my data. So I had to do a lot of detective work to figure out exactly how many eggs had been laid, sort of using those partitions and the pollen provisions that were left over. And so in 2021, I was able to open them up prior to the emergence of those, I think they're Maloidae blister beetles. And so no harm was done. I was able to just get them out of there before they could go to town on all my data. And so I mentioned that we released Osmia lignaria as a focal species, but after 1 year of data collection, we were kind of curious, what if we just put empty tubes out, would cavity nesting species nest in them? And so in 2021, like I said, we released Osmia lignaria. You can see the little deployment capsules we put together out of PVC, and they emerged right here underneath the nest, and sort of using pheromones and based on proximity of these available cavities, nested in them. We got some okay data. In 2022, we did the same, released Osmia lignaria, but we also set out empty tubes in varying sizes for the various different types of cavity nesting species that we know are likely on the landscape, and it worked pretty well. So in 2023, we only released those cavity nesting tubes, and same with 2024. And here's kind of a distribution of what those T-sticks looked like. We had tubes varying from 4 millimeters to 10 millimeters. Even though the 10 millimeter ones are for carpenter bees, which aren't known to be on the landscape, we figured we'll put them out and just make sure, since we're not totally sure what bees are present here in Washington anyways. And like I said, we had some success in the community nesting tubes, but I have a graphic here for you on just the blue orchard mason bee data. Since there was a bit more nesting success in those first 2 years, and I just haven't had a chance to update these graphics. So looking at 2021 and 2022, this is the number of eggs that were laid and successful development into the pupal stage. And so in 2021 in the yellow, what really stands out is that the youngest burn, the 25-mile creek, or the Cougar Creek fire, did super well and had a lot of nesting success compared to the other burns. Interestingly, the control did quite well in 2022, which kind of leads me to believe that maybe the landscape has shifted so far out of its historical fire regime state that it's a totally different ecosystem completely. It's no longer a ponderous pine landscape. Maybe we're looking at something different. Maybe we're looking at something with a lot more ceanothus and fuel and floral resources. Maybe we're looking at something with a lot more mixed conifers, so there is more cavities because there's just more wood. I'm not really sure, but I was surprised to see that in 2022 we had such high nesting success at the control site. I will say in 2022, these numbers here in the green may be artificially decreased due to the fact that once again, the predators had their way with my data, and a lot of it was consumed by birds. So you could see where the birds had pulled the tubes out of the nesting boxes and eaten all the insect larvae and pollen inside. And so again, a little detective work was done. Some of them were still intact, but may not necessarily be reflective of how successful the bees were there. 25-Mile Creek was not a site in 2021 because it burned in 2021. And in 2022, we interestingly had no success. So 1 year post fire seems to indicate that maybe that's not enough time for those cavity nesting species to recolonize from edges or wherever they're located. Or perhaps there were so many cavities on the landscape that were better than my artificial PVC houses that they were just not interested. It's hard to say. So I mentioned the partial consumption by birds. In 2023, we put together these really fancy bird guards. We put them out halfway through the season when it looked like there was some activity, so that the bees hopefully wouldn't be dissuaded from beginning to nest there. And it seemed like it worked okay. We had no predation in 2023, so patent pending on that one. After we take the tubes back to the lab, for Osmia lignaria, it's easy. We know what their pupae look like, and we can cut them open and we can quantify them. But for these other species that are present on the landscape, it's harder to know what species of bee it is nesting in those community tubes, just based on the pupae. Maybe there's someone out there that can ID just native cavity nesting bee pupae, but I certainly cannot. So in order to bypass that, we overwinter them artificially in the growth chamber in the lab, basically send them through a rapid cooling system and see if we can get them to emerge as adults in like an artificial spring that we've created so that we can then capture them and identify them so we know what nested there. And that worked pretty well last year. Here's some of the examples of the pupae that we were able to put in there. So we had at the 25-mile creek fire in 2022, which I mentioned didn't do so hot in 2021, we had a lot of small, what appears to be osmia that nested in the tubes. And we were able to get to successfully emerge. We also had a lot of, I made graphics for this. So Osmia lignaria on the top, we're familiar with. Here we have a leaf cutter bee, likely something in the genus Megachile, that uses leaves to create its pupal cases, overwintered those. And then, like I said, there's these small osmia as well. So when analyzing this, same sort of infographic as before, but this time we're looking at for each cavity nesting bee species, oviposition and larval development as a function of time since burn, fire severity, and kleptoparasite density, because if there's a lot of kleptoparasites, it's likely that not a lot of those eggs were able to successfully develop into pupae, and I imagine that'll have a direct effect on their fitness. And those kleptoparasites came from the field, not the lab, right? So it's probably reflective of what's happening in the field. And same thing, adding additional environmental variables where possible, because in ecology, nothing's in isolation, always in the system. So trying to add that in if there's power, and using space as the random effect. We can't use time in this analysis like we could in the previous analysis because I don't have temporal data on when the tubes were filled. Instead, they're left out for an entire season. So it's just a metric for the year. Because I'm not necessarily checking like, oh, 2 eggs were laid in April, 2 eggs were laid in April, 2 in May, something like that. I'm just collecting for the whole season. So we can only sort of compare by location, by burn site, by space. All right, my final objective. This kind of speaks to your question about floral resources and how to quantify that. We plan to use next-gen sequencing in order to determine which plants the bees are visiting based on the pollen they've collected. And my hypothesis is that bee community metrics will be affected by time since burn, fire severity, and the species richness of the forbs, woody shrubs, and flowering trees on the landscape. So this, of course, is tied to fire severity and time since burn, because we know there's more floral resources post-fire. But the assumption is that the more options the bees have to visit, potentially the higher their diversity, abundance, et cetera, will be, tying back to that sort of first question that I asked. So in order to collect data for this objective, we sort of have 2 to 3 data streams. The first is that I'm trying to glean pollen from the soapy water in my traps. So in this case, we know that the bees were out foraging, they fell in my traps, they probably had pollen on them. We're not able to tie that pollen that we strain using very, very small filters to a specific bee because they were all together in the trap, but we can tie it to a site and to time from when we sampled. So trying to collect pollen that way, not well represented in the literature, very like preliminary thing we're trying out, and unclear at this time if it's been successful, but if it is, then that would be a really wonderful day to get site-specific pollen data from each site through time using destructive sampling. The alternative is to take the pollen directly off the bee. So where I did see pollen on bees in the corbicula, which is the pollen carrying basket, and scopa, which are pollen carrying hairs, I used a sterilized probe under the microscope and sort of like scraped it off very carefully. There are ways to do that with a centrifuge, putting bees directly in the centrifuge, which would have been easier. So, and then from objective 2, obviously where the pollen provision hasn't been consumed by the egg, and so there was no successful development into pupae of the insect, we're able to just take those pollen provisions right out of the tube. Again, we don't have time-specific data associated with these pollen provisions, but it is quite a bit of pollen that we know didn't just like fall in from a tree or get blown in by the wind, and it's quite a lot. So this is great data, but of course there's not that many pollen provisions that weren't consumed by the larvae, so it's kind of a matter of trying to get pollen where we can from our different previous objectives. And once we have that pollen, my plan is to do DNA metabarcoding on it. I'm going to use the ITS2 region in order to do that. And ideally, it would be great to determine a sort of short list of plants that are returned that we know the bees are visiting in these post-fire landscapes. I'll also be comparing the relationship between the results we get from the DNA metabarcoding to the results I collected in the field, and see how well the sort of 10 by 10 random plot transect survey reflected what the bees were actually visiting, reflected in the pollen that they've collected. So it could be an interesting sort of check on field-based methods for floral quantification by doing this genetic work. And once I have that, I'll run a GL generalized linear mixed model again, look at, like I said, this is going to look very similar to objective 1, pollinator abundance, species interest diversity as a function of time since burn, add in those environmental variables where we can, random effects of space and time. But then we can add this grouped plant species richness as a sort of effect that could influence what pollinator abundant species richness and diversity looks like, with the idea that if there's more plants and more resources available for them on the landscape by site, we'll probably see more bees and more different types of bees. We'll also examine changes in forbs, woody shrubs, and flowering trees through time, because we'll have this temporal data provided that the trap pollen collection functions, and ideally create a list of high priority plants for pollinators through time, according to fire severity for land managers. So that's kind of where I'm at so far with this project. Still a lot of data analysis to do. It's still a lot of data processing to do in terms of bee identification. And then just starting in the genetics lab to sort of do that pollen work and do a check on how my collection efforts have worked. I do plan to do another field season focused solely on the cavity nesting bees and pollen collection this summer. So there is some opportunity sort of to pivot if it turns out that collection from those soapy water traps didn't work. But science communication is really important to me despite that. So thanks for inviting me here. And I'm also going to be at NSOC in 2024. There's a piece in the Seattle Times you can check out that sort of highlights what I've been doing, which was a really exciting opportunity and a way to sort of like bring this out to folks. I feel like whenever I talk to people about my research, I'm always like, I'm studying bees and fire. And they're like, oh my gosh, yes, I love honey. And I'm like, no, that's not at all, like not even a little. So it's great when we can sort of get away from the honeybee a little, despite its importance, both economically and agriculturally. Native bees are the best, especially in terms of resiliency to climate change and just supporting our native biodiversity that's here. To that end, I've met with one specific, but I'd like to meet with more local native bee suppliers. As I mentioned, Osmia lignaria is used for pollination in Eastern Washington a lot, mostly for cherries and other fruit trees. And there's a lot of interest in what other cavity nesting species I was able to get to nest in my traps to see if those could be used in an agricultural setting as well. And then I'm hoping, to finish this project up, the first objective, at least in 2024. So a lot of my effort right now is going into identification in the lab for that first objective. In terms of looking forward, like I said, a lot of analysis and writing to do, another field season planned in 2024. I think that we talked about food security, and it's important to think as well about indigenous foraged foods located in natural forests, where those tribes that historically resided there do still have foraging rights. And so for plants like huckleberry and other native berries that are still foraged and consumed by humans, the native pollinators that are out there are critical for that to be sustainable. Of course, important for conservation of both the plants and the pollinators located there. And as we see increasing fire severity and annual area burned, I think that it is critical to know how that's going to be affecting our native pollinators moving forward. So thank you so much for your time. A couple of folks I want to thank listed here, and I would love to take any other questions in the 10 minutes we have left. Are you going to ask a question? Yeah, please.
When you deploy your little like cavity nests,
Yep.
have you been able to collect pollen directly from those nests, or is pollen present even after you're taking them and the loading of like everything?
Yeah, great question. So I'm going to back up just a little. Yes, I can take pollen from there, but only where, if I was to take all the pollen from there, it would take data away from objective 2. So it's hard to collect data for both objectives at the same time, because then the egg wouldn't have, I'd have to do it early before the egg had the opportunity to consume the pollen. And then we couldn't use that tube as a measure for fitness, because it wouldn't be like reflective of, though our measure for fitness is a proportion of eggs laid to successful larva development. So if I took a whole tube, that would just be a tube that I couldn't use as data for that other objective, which, though I've had success in the mixed community sort of analysis for cavity nesting bees, it hasn't been a ton. So I'm hesitant to destructively sample from that data set, given that there's not a ton there to work with already. But that is like the most successful way I've found to collect pollen, is where the egg doesn't successfully develop and doesn't eat the pollen provision just because it didn't, or whatever, there wasn't an egg laid there, or it didn't have the resources it needed, or whatever the case may be. I am able to take that pollen that's left over once they've already moved through. Yeah, but it's tricky.
Right.
Yeah.
You end up getting all the pollen.
Yeah. And if they don't, the beetles will. So you've got to get in there right at like the perfect time. Yeah, everyone wants to eat pollen. It's a great sugar, carb snack, protein. Thank you. Yeah. Yeah.
So when you had your, like your tube with eggs that you wanted to identify a species you put through, so that you pulled them down. So you could
Mhm.
get that to work.
Yep.
So I guess I'm thinking, is that similar to like germination for plants? Like, is it the thing that activates them
Mhm.
for continuing to grow and to eventually emerge only temperature based? Is there like a set gestation time before that can occur?
It's very similar to plants exhibiting dormancy, where they sort of need like a chilling period and then a warming period. A lot of insect development is predicted using degree day models that are purely temperature inputs. It's not like, well, in terms of photo period, we can also mimic that in the lab. So in our growth chambers, we have a set amount of day and night, which was reflective of like what it would be when it's that warm / cold. But we're basically able to just do like exactly that degree model, that heating, that cooling and heating, but under a decreased amount of time. And it is mostly just temperature. It's not really, I'm trying to think of other indicators that might cause them to come out of their pupal cases early, and really temperature is the main driver of their development. And that's why local beekeepers that are using Osmia lignaria, blue orchard mason bee, are able to sort of control their development and get them ready to emerge directly when they sell them to the growers, so that the growers can just put them out in their orchards and they're ready to go. So it's that like that piece that can be easily manipulated in a commercial sense as well. So yeah, it's kind of, they've got large refrigerators just full of pupating bees that they're keeping inside until they're ready to send them out into the world. So it is, yeah, it's pretty neat. Yes.
Have you caught any like aphis honeybees, like from nearby,
Mhm.
like either, people who have set them out for things, or like
Yeah.
different, and like what percentage is that making up a large percentage of what you're catching, or is it really
Yeah.
1 or 2?
Yeah, great question. I have caught Apis mellifera. I catch about 5 to 10 a year across the whole season, so not a lot. It's not surprising that I'm catching them, because I mentioned 500 meters is the average native bee flight range, but Apis mellifera, as you know, can go for miles looking for pollen. And so there are some orchards sort of bordering the national forest at pretty much all of my sites. If you've been to Chelan County, you know it, there's a lot of fruit production. And so, yeah, luckily not too many. And honestly, I'll probably not include them in my analysis because they're not really a good metric for pollinator health on the landscape if we're thinking about native bees. So I'll probably just remove them. But yeah, not too many. Which is
I
good.
guess you're getting so few that it's not like competitive pressure at that number. like,
Yeah.
At that density, right? Yeah.
I don't imagine so. What I do get a lot of is osmia. And osmia is really difficult to identify, as you probably maybe already know. There's not good keys for the West at all. There's an expert in Logan, Utah, who seems to know what species are here, and otherwise that information is really not available. But I'm fairly certain I do have a lot of Osmiolignaria, which it's hard to say if that's native Osmiolignaria or released Osmiolignaria from the blue orchard mason bee from the orchards. But I would say that would be the competitive pressure from agricultural settings, if any. Yeah. Good
Can I
question.
ask a question about the model?
Yeah.
Uh,
Yeah, please.
How does the model account for the nonlinear relationship between years since burn?
What do you mean by nonlinear relationship?
Is it, I'm guessing like it peaks at like a few years after the burn and then
Mhm.
maybe it tails off
Yeah,
at the,
through time.
Yeah.
So I was hoping I'd get some tough stats questions from this group, and I'm glad that you're going to ask me a stats question. No worries. I think instead of looking at like one site through time, it'd be a really long-term study to look at, say, the 25 Mile Creek fire from 2021 when it burned all the way until it's 50 years post-burn. But because the landscapes are pretty similar, I think instead I'll be looking at like sort of linear relationships, but in that snapshot of 1 year, and then trying to tie my sites together. Of course, there's some variation, but for the most part, they're all at the same elevation, same aspect. Most of the slopes that they're on are facing southeast, same direction, all ponderosa pine dominated. Really, when we selected sites, what we tried to make differ between them was just the year since burn, and tried to keep everything else as similar as we could, given it's an ecological study and variation is inherent. So that said, I'm hoping to just look at the linear relationship between, like, 2021 burn in 2022, 2021 burn in 2023, but then I'll have, 1 year burn, 2 year burn, 3 year burn just from that fire. And similarly, I'll have 3, 4, 5 from the Cougar Creek fire, and I'll have 6, 7, 8 from the Chelan Complex fire. And then that sort of, 1968 control burn is sort of like the other end of the spectrum. Like, how is it different, if at all, especially given it's so close to where the Chelan Complex fire burned. So kind of doing it that way instead of accounting for how it's changing through time, if that makes sense. Yeah. A little bit of an easier way to do it too, because it would be really hard to look at one site through 3 years and see that curve. But it would be a great study, but it would be a long PhD. I'd probably be here for a while, longer than I want to be. Great. Any other questions? Thanks for your questions. I really appreciate them. Not really solid questions. Great.
yeah, okay, if there's
Great.
no further questions.
Yeah.
Thank you all for coming to QuantSab. Oh,
It's gonna.
okay. Hope to see you next week. That's all I have to say.